Hi, welcome to Murphy Microbiology. I am Dr. Laura Murphy, and today I'm going to be doing a demonstration of what is known as the smear in microbiology. This is one of the first steps that is important to get mastered prior to doing most of the stains that we see in the microbiology lab. So to get started, you want to make sure you have your Bunsen burner, some sort of staining tray, and microscope slides, a culture, either liquid or solid. Today we've got both the liquid culture and we have a solid culture growing on a plate. Some sort of transfer implement. I prefer to use the loop, and then of course a marker to mark up your slide. So getting started, when I teach my general microbiology students and they're starting off with making stains and smears, I like to suggest that they draw with some sort of permanent marker a circle onto the slide that's going to give them an area in which they're going to be placing their sample. And this is just kind of a guideline for where they should look once the staining is complete. So once you have drawn that circle onto the slide, you want to invert it because your actual organism is going to be placed on the top. So again, starting off, I like to use the loop. We always have to sterilize our transfer implements in microbiology so that we're not contaminating what we're doing with microbes that could have been from the air from a previous experiment. I'm going to demo today using a liquid culture. So to do so, you want to get at least one loopful of your sample. And I'll talk about this more in a moment. And once you have that, you're just going to gently rub it within that circle that you drew earlier, okay? Kind of spreading it out as you go. Once that is done, you can sterilize your loop again. Now, a couple of things to note here, and this is how you get good at doing smears. You want to make sure you have enough cells on your slide so that you're going to be able to visualize them later, but not too many so that they're clumped together and then you can't tell what you're looking at. So with this particular culture that I have here, it's fairly turbid or concentrated. I think one loopful of this is plenty. If it were a dilute culture, I might add a second loopful to the slide and I would just repeat the protocol I showed you. If you are starting from a solid, it's a slightly different procedure in that first you're going to use your loop to put one or two loopfuls of water onto the slide and then take your sample and mix it in to that circle. That's the first step of a smear. The second step is critical, and this is that you have to allow all of the liquid on that slide to dry before moving on. For the sake of time, I'm not going to sit here and wait for this particular slide to dry. So I have another one over here that I've already air dried on its own. There is no more liquid on this slide and I can proceed with my smear. The second part of this is going to be what's called heat fixation. Heat fixation is passing that smear through the flame so that we can not only kill whatever's on that slide, but also adhere the cells to the surface so that when you come back and do any of your staining techniques, you're not gonna have a problem with washing off your cells. So to do heat fixation, you can hold the slide in your hand or with a clothespin, and you're just going to pass that smear that you made through the flame, back and forth, 3 times, taking a short break in between each one so that you don't overheat the slide. And now you're ready to move on with whatever stain you're interested in.